Lovely. Welcome, everyone. It's good to see so many of you. So let's see. I will still be admitting people as we go, but welcome to Foresight's Molecular Machines Group. This is now a follow-up presentation to a presentation that we had a while back where William Shih presented his work. And many of you afterwards have kept on nudging me throughout the months to please, please, please have William back when he has the results shared that he left us hanging on a cliffhanger with at the last presentation. So here we are. I'm really, really, really excited to have Will back. Thank you so much for for joining on such a short notice. I know that people are really looking forward to learning how research has progressed since then. I will share much more information about you in the chat, including the last talk that you gave with a seminar summary. So if people want to get up to speed, then I propose you look there. But if people want to enjoy the presentation, then I propose you look on the screen. So thanks a lot, William, for joining. It's really a tremendous pleasure to have you. And we're super excited about your research. You will be presenting for maybe 35, 40 minutes. Let's see, maybe we get through it faster. And then we'll have, about 15, 20 minutes for Q&A. So I would recommend to everyone, please start collecting the questions early, because at the end of it, we're always running short on time. And so if you want to get your question heard, then start collecting them as early as possible. Okay, without further ado, William, I'm terribly excited to have you back. Welcome. And I can't wait for the Q&A.
Great. Well, thank you, Allison, and everybody for inviting me back. It's my great pleasure to share with you a progress report on our crisscross self-assembly. We're very excited about this method, and I hope to convince you that we've made a big leap forward in self-assembly based nanofabrication. So I changed the title a little bit to the title of the manuscript that we're working on. That our current title is Prototyping Multi-Micron Crisscross Structures from Combinatorially Assembled DNA-origami Slats. And hopefully that will make more sense by the end of the presentation. The main protagonists of this story are a trio of talented young investigators working in our group, Chris Wintersinger, Dio Minov, and Anastasia Urshova. They all were graduate students. Actually, Dio graduated, but is continuing on in laboratory as a postdoctoral fellow. And this is also in collaboration with these other co-authors, some of them in Peng Yin's lab. And I'd like to frame this by, one could imagine, a grand challenge in nanofabrication. So can we grow finite addressable structures the size and complexity of biological cells? At the lower end of the spectrum, it could be E. coli, which already would sound mind-blowing if we could do that. And at the bigger end of the scale, we could say eukaryotic cells, something like a human cell. And this all seems very, very far off. But what I'm here to tell you today is that it's not that far off, at least in terms of building a static structure about the size and complexity of a cell. So I don't want to make it sound like we can make something of the functionality of a cell, but at least even to make something of the size and complexity, a static structure the size of the cell sounds like science fiction, but that's no longer the case. So at least I hope to convince you of that. So the starting point is DNA-origami. So this was a big event for DNA nanotechnology developed by Paul Rudiman in 2006. And here you have this 7,000 base genome, typically of the M13 bacteriophage. We know what that sequence is, and based on that known sequence, we chemically synthesize hundreds of oligonucleotide staple strands that are 20 to 60 bases long that are programmed by Watson-Crick complementarity to pinch together this long scaffold into this parallel array of double helices. And this is a really robust method. It works extremely well. It doesn't matter if you're a specialist, if you're a non-specialist. It's very, very robust, high yield, and easy to do. And it's become very, very popular in the field. Here's an example of the iconic disk with 3 holes, atomic force micrograph, 100 nanometers by 100 nanometers by 3 nanometers. And I like to say it's like you have 2 ribosomes worth of molecular silly putty that you can mash into any kind of shape that you want, but it's scalable self-assembly, so you could easily be making trillions of copies of this per droplet of liquid. So quite amazing. But then we come back to that question I raised earlier of, okay, that's cool. And for many applications, that's big enough, 2 ribosomes. But sometimes we want to make something bigger. And it's kind of like the difference between if you wanted to assemble a ribosome versus you wanted to assemble a cell. Sometimes you want to make cells. Sometimes you want to make pocket calculators. Sometimes you want to make smartphones. So we have this constant urge to try to make bigger and more complicated objects because that oftentimes opens up new opportunities. And we could ask, what would be the utility of building larger things? And the last presentation I gave, which is on the YouTube channel for Foresight, thanks for uploading that. And so you guys can see that one motivation for growing very, very big structures is for the purpose of amplification. And we're specifically very interested in trying to develop ultrasensitive diagnostics that can take very specific detection of tiny, tiny analytes, and then based on recognition that that analyte is present, grow these large structures that might be millions, billions, trillions of times the mass of the thing we're trying to detect. And in that case, it's very important that we can amplify. The exact shape that we build is maybe not that important. It's just that we can reliably grow this thing. And maybe even more importantly, that we can prevent anything from growing when there's nothing there. This turns out to be a challenge in self-assembly. How do we program something to grow that's thermodynamically favorable, but then control it? Have kinetic control so that this process only starts when we want it to, despite the fact that it's thermodynamically favored to just everything to grow. And the second application, which I'm going to be sharing with you today, is, well, what if you want to make big things because you specifically want to make big things? You want to make devices where the size and shape is important for the functionality that you're trying to achieve. So let's call that for now microscale shapes with nanoscale features. You could argue that cells are something that is in this category. And here's some electron micrographs, a transmission electron micrograph of the flagella organelles from Chlamydomonas algae. And here's a scanning EM of the algae. So these are amazing organelles that are a couple hundred nanometers in diameter and roughly about 10 micrometers in length. So something much, much bigger than a ribosome. A ribosome, by comparison, is about 25 nanometers per dimension. And there's this intricate organization within these flagella. Again, the idea that people could fabricate something of this kind of size and complexity might seem very far off. But in principle, we would hope to develop methods so we could match this kind of complexity. And why stop there? Why stop at growing something the size and complexity of an organelle? Why not try to build something the size and complexity of a cell? Or if you want to pick a different metaphor, why not build something that is a microscale robot with nanoscale features? Or why not build some kind of wet molecular computer that has elements that communicate with each other over the length scale of multi-microns? So an analogy being, if we look at things like integrated circuits, what can you do with the integrated circuits that has a relatively small number of transistors versus one that has millions or billions of transistors? So we're always interested in trying to scale to greater and greater complexity to get to that, when needed, get to those larger degrees of sophistication. And as background, there's towards this goal of trying to build very, very large structures. The paradigm that I'm going to be discussing today is hierarchical self-assembly. So can we start from DNA-origami, which works really, really well to make something about twice the size of a ribosome? And then can we reliably get those things to hierarchically self-assemble into much larger structures? And to do that in a very reliable, robust, high-yield fashion. And so this is a really nice paper from Gregory Tikhomarov and Lulu Chen at Caltech. It was published in Nature in 2017. And what they did here is they programmed, in this example, 64 different DNA origami that have roughly the same shape, but a different specific pattern on them. And then they're programmed with specific glues on their edges, such that if they hierarchically mix these things together, let's say 4 at a time, to make a 2x2 array, and then take those and then mix those together to make an 8x8 array, a 4x4 array, mix those together to get an 8x8 array. Then on the one hand, it's pretty amazing that they're able to generate these structures where every single element in this object is independently addressable. That's pretty remarkable. It's approaching the size of an E. coli bacterium. But then maybe it's not quite ideal for applications where we want to have high yield, because if we look at the yield of these structures as a function of how large they're trying to grow them, you can see there's a precipitous drop off of the yield as these objects get larger and larger. So this 8x8 array, according to the metrics, has something on the order of 1 to 2% yield. So we have a situation where there's a disconnect between the way they're making the individual building blocks, these DNA-origami, which is a very robust procedure. You can be kind of sloppy, just add in a large excess of your staple strands, and then it just works and you have very high yield, versus trying to assemble these individual origamis into structures. Now you have to be extremely careful about preparing each one, making sure the stoichiometry is exactly correct, fussing around with the interfaces, making sure the interfaces are very correct. So it requires very high precision, and it's not robust at all. And at the end, as you try to make larger and larger structures, then you start running into these yield problems. So a question that one might ask is, well, why is it that DNA-origami itself, which is made from 200 components, works really well and is robust, but then trying to combine 200 origami into a larger structure doesn't have that same robustness? And one thing you could immediately jump out and say, well, the origami themselves, they have this long master strand, this scaffold that is directing the action. And it's that long master scaffold that's giving us that robustness. But we don't have access to a scaffold that's long enough, robust enough, that it's basically big enough to reliably scaffold this large structure. So we could take chromosomes that are quite long, but it turns out DNA, once it gets that long, is very hard to handle. It's very fragile. You just sneeze at it and it falls apart. So for a bunch of practical reasons, it's not reasonable to try to do nanofabrication with these extremely long strands. So then the question is, okay, is there some way to get the kind of robustness and control of DNA-origami, but leaving out that long strand? And that's the method. Well, we've come up with a method that we think does exactly that, that we think is key for allowing us to achieve very, very high yield, or relatively high yield of these larger structures. And I'd like to point out another piece of background research. So the work I'm going to be telling you about is using building blocks that are these DNA nanotubes, 6-helix bundle nanotubes that are about 450 nanometers in length. And in parallel to the work that our group was doing, Hao Yan's lab was doing something similar. And so they published this paper last year in Nature Chemistry. And they came up with a very nice name. They called them meta-DNA structures. And so the idea here is that you have, what if you imagined conceptually each one of these DNA nanotubes, which itself is almost a half a micron long and about 7 nanometers in diameter. What if you think of it analogous to a DNA strand? And you imagine it's this modular connection of little adhesive glues that, in this case, they're acting like artificial base pairs. Whereas with natural DNA, you just have 4 letters, A, C, G, or T. But in this case, we could decorate these DNA-origami with some library of sequences. We're not restricted to just 4 sequences, and they could be much longer than 1 nucleotide. But functionally, they're behaving like specific weak glues that we can make orthogonal to each other. And so they could program these things to wrap around each other to make something like a meta DNA duplex. They could get them to fold into different structures. So this is very similar to what I'm going to show you now. But the added ingredient that I'm going to show you is we've come up with a system that has absolute control over nucleation in a way that's kind of like having a scaffold there, except there's no scaffold controlling the entire process. Instead, we have a small seed that is absolutely needed to initiate the growth, but the seed itself is very small compared to the final assembly. So here's our building block. It's also a 6-helix bundle that's about half a micron long. And what we're doing is we're programming it with staple strands that are coming out at 14 nanometer intervals along this object. And at each of those 32 positions, we can put any sequence we want. So it's kind of like we have that meta base pair sequence at each of those 32 positions. And the examples I'm going to be telling you about near the second half of the talk, we're using a library where we have 32 possible sequences, well, actually 64 possible sequences. So it's 32 pairs of possible sequences at each of these positions. So with regular DNA, you have 4 possible bases at each position. With our structures, we just chose to have 64. And that's kind of an arbitrary decision, but we found that 64 is a good, it works well for us. And we're programming these structures, we're limiting them to interact at a roughly 90-degree angle. And there's a reason that helps us to enforce this absolute C dependence. The interaction has to be pretty weak so that at the temperature of assembly, when things come together with just one bond, then it's relatively weak and it's transient, it's going to fall apart. And that turns out to be important. So here's the overall architecture of the growth of at least one-dimensional structures. So again, we have these half micron long DNA nanotubes. They're about 7 nanometers wide. And in this case, we're only using every other handle position. So we're just using 16 of those 32 positions. And this is the structure that we're trying to grow. It's propagating from lower left to upper right. And it's kind of like beads on a string. We are running this self-assembly at a temperature where, in this case, you need to make 8 bonds for stable attachment. So we just raise the temperature to the point where only 6 or 7 attachments maybe is not quite enough energy. So for example, if a golden vertical slat tried to come in at this position here, it can only make 1, 2, 3, 4, 5, 6, 7 bonds. And so at this temperature, it's likely to fall off before the next thing happens. So let's say it falls off. But if we look in the horizontal direction, we can make 1, 2, 3, 4, 5, 6, 7, 8 bonds. And then look what happens. It's now created new binding sites for the next set of vertical ones. So in this example, there's, let's say, 16 unique horizontal slats. So each one has a different meta sequence. We picked a different sequence at each of those 16 positions. And then let's say 16 vertical slats. So this is a periodic growth where the entire structure repeats every 32 slats, 16 vertical, 16 horizontal. And the important thing that we're trying to engineer is absolute C dependence, so that nothing will get started except when we want it to. Maybe I should say a little bit about why that's so important, even for fabrication, because you could imagine, what if we're trying to scale to something like 1,000 different building blocks, and each of those building blocks has the ability to seed nucleation on its own. So you could imagine the kind of potential chaos of adding all 1,000 slats, because now you get a lot of consumption into these little microstructures, and then you might get depletion of the building blocks before complete things get built. And furthermore, now you have a purification problem. You have to get rid of all these incomplete assemblies. So it's going to work much better if you can only grow things when you provide a seed, and then it's all or nothing growth. You either make it or you don't. And then it's going to be functionally very similar to DNA-origami, which there, again, the strength is it's all or nothing growth. You either build it or you don't, depends on the scaffold, and you could have a huge excess of the building blocks, no problem. So here we're looking at the birth. So we're trying to understand how is it that this kind of architecture allows us to prevent unwanted spurious nucleation. And we can understand that from a free energy diagram, where free energy is on the y-axis and the growth coordinate is on the x-axis. And we can see early on in the assembly of these structures, because we're running at a temperature where you need 8 bonds for stable attachment, that when you're only making 1 bond, that's not enough to pay the entropic penalty of bringing this thing in. So you're going up the energy curve. It's very unfavorable. And as we continue this very improbable growth trajectory, it keeps on getting worse and worse. So as we add more and more slats, you're starting to make more bonds, but it's still less than 8. And so every time you make less than 8 bonds, you're going up the energy trajectory. So it's getting exponentially more and more difficult. It's like climbing Mount Everest without an oxygen pack. It's getting tougher and tougher with each step until you get to the point where you're at the peak. That's and we can engineer where this peak is. In this case, it's 8 horizontal slats and 8 vertical slats. And once you can make it to the peak, now you can start going downhill. Now you can start adding with 8 bonds at a time. But the important thing is it's going to be very, very difficult to get up to that peak. So the rate at which you cross the peak spontaneously is going to be very slow. And perhaps more importantly, if for some reason it was happening faster than we wanted to, we could just redesign this critical nucleus to require an even larger number of slats. And it turns out, for technical reasons, every time we add a pair of these slats to this critical nucleus, so if we go from 16 to 18 slots, for example, that's going to slow down the rate of spurious nucleation by another factor of 1,000,000 or more. So the point is, and then we can keep on doing that over and over again. So what I'm going to eventually tell you about is not 8 plus 8, but 16 plus 16. So you can get to the point where it's basically the odds against forming this within a certain temperature range is astronomically low. It really should not be happening in theory, even if you had Avogadro's number of components and waited 10 to the 18th seconds or whatever the lifetime of the universe is. That now buys us the opportunity to control the growth. So the gray curve represents this hypothetical situation that is effectively never going to happen. And then now we could program a tunneling through that energy barrier by providing a seed that's going to get through this energy barrier. And in this case, we're using architecture also from Hao-Yan's lab that they called the gridiron DNA origami. And it turns out the spacing of this structure perfectly matches what we want to do. So we found it very convenient to adopt the gridiron from Hao-Yan's lab. And so what we're doing here is we have this adapter set of horizontal slats that's now interacting with the seed with very strong base pairing interactions. So this is in distinction to the way the slats themselves interact with each other with only very weak bonds, because we want them to fall apart instantly. But in this case, engaging with the seed, we program pretty long base pairing interactions. So you get this robust, non-cooperative, independent attachment of each of these horizontal slats to the seed, so that when you form the seed, then now you basically paid for the entropic fixation of these blue elements with a huge number of base pairs, or pretty large number of base pairs. And then now they're fixed in high local concentration so that they can recruit these vertical slats. And then now you've basically created something that looks like the end of a ribbon, and the growth can start taking off. You can start going downhill because you're making those 8 bonds with every addition. And for periodic growth, then it'll proceed like so. And so here's some data. These are negative same transmission electron micrographs. This is the gridiron DNA origami seed. And then here we're just showing one of these adapter slats docking to it. Here's with a much larger number of slats that's docking to it. In this case, we're programming, we could program it to have different slats attached at each position if we want to, which is the way that we want to do it if we're using it as a seed. So in this case, you can kind of make out that the seed is behind here. It's kind of a little bit low, a little bit faint, but it's binding 8 of these horizontal blue slats. And then those 8 horizontal blue slats are binding 8 vertical yellow slats. And then the process gets off and running. You can make these periodic ribbons. And at least to the limit of detection, if we run this at a high enough temperature, close to the reversible temperature for growth, then we just don't see any spurious nucleation, which is exactly what we want. We want the all or nothing seed dependent control of DNA origami, even though our seed is much smaller than the structure itself. So it's the spiritual advantages of DNA origami, but with something much smaller, proportionally much smaller than the scaffold. So we think this is really great. In this particular design, there's no angle control, so the structures are kind of floppy, and that's something that could be useful or that we could potentially engineer around using different ways. So one of the ways is by adding in additional elements to further rigidify. So with this initial design, there's a lot of potential flexion, but one thing we did was to add in a third layer on top and then create a triangulated structure. So in this case, this is an object that's much more rigid. So there's and here's a 300 nanometer scale bar. So the main point here is twofold. One is that we're not limited to only growth in this one-dimensional growth. As I'll show you, we can make 2-dimensional growth, and we can also grow in the Z direction. Because in this case, it's only three layers, but it proves the point that we can also grow by stacks as well. So we can really achieve, as I'll show, or at least I'll argue, that we should be able to achieve 3-dimensional growth using this kind of design language. And the second point is that we can rigidify these structures using tricks like triangulation, kind of common sense strategies from simple mechanical considerations. All right, so a big development in this method is when we decided to switch over to the impressive level of nucleation control with the 8+8 design. And then we switched over to a ludicrous level of nucleation control with this 16+16 design of the critical nucleus. So here we're using every single one of those 32 positions on this 0.5 micron long building block. And when we do that, we found that the process just started to work better and better. In fact, we were really impressed how well this method works, and I hope to impress that upon you as well. Now, it doesn't work infinitely well, but for a first try, we're just really excited about it, and we believe that it'll be amenable to a lot of optimization. So this is really just a progress report. So here's the structure now with the twice the density of those binding sites. Here's a 200 micron nanometer scale bar. So just drawing back to that example of the flagellum from the Chlamydomonas algae, so that thing was about the same diameter as these ribbons. And we can grow these ribbons comparable length to those flagella. So those flagella are about 10 microns. We can routinely grow these periodic structures at least 10, 20, 30 micrometers long. So we're really, from a dimensional point of view, for the first time, we're creating these kinds of addressable structures comparable in size to organelles, such as a flagellum. And so I'm going to now go through some more examples of these periodic structures that we've built. This is just to impress on audience members that these are addressable structures where we have they're periodic in that we have a 16 plus 16 slats in the asymmetric unit that has dimensions of about 220 by 220 nanometers. But within each of those repeated blocks, it's fully addressable. So we can create intricate patterns within each of those repeats. So here we're using DNA paint, and this is a method of super resolution microscopy. You can image below the diffraction limit of light. And what we do is we put sequence specific handles at different positions that allows us to basically image objects. So here it's supposed, the imaging, we're still working on it, but it's supposed to say Wyss, like Wyss Institute, which is where one of the places we're working. This one is supposed to be a smiley face, and maybe it's not so clear that it's a smiley face, but hopefully you're convinced that this is not just some random thing that we're imaging here. But the point is that they're addressable. So what I've shown you so far are these structures up on the top that are about 300 nanometers in width, and then we can make them very long. So these are just a couple other kinds of designs of these 1D ribbons. So in this case, what we're doing is we're programming just one side of the slats to attach, and then creates another face like so. We call this zigzag growth. That's what we're currently calling it. That allows you to make ribbons now they're more like a half micron in diameter by using this architecture in the middle. And then on the bottom is a structure where it's another kind of zigzag growth where it's basically going, in this case, right, up, right, down, right, up, right, down. Again, with this notion of we're just going to be binding 16 slats at a time, where we're just using, let's say, the left face or the bottom face. And so you're creating this, in a sense, single layer interfaces that are stacking in different directions. And I should mention that in these cases, we're programming bidirectional growth. So we're growing both to the left and to the right. In the future, we'd like to scale to even larger number of growth fronts that will allow us to get faster growth. But for now, we're focused on these bidirectional growth fronts. So we can also get two-dimensional growth. So I'm not going to go into great detail of the exact architecture here, but let's just suffice it to say for now that it's possible to arrange these binding sites in a way that you can get two-dimensional growth. So again, we have this repeat of 16 plus 16 slats, so about 220 by 220 micrometers, but now that's repeating in 2 dimensions. So here's a 10 micrometer scale bar, and you can see there's something growing on these TEM grids, and the dimensions are something like on the order of 10 microns. So this is about the footprint of one of those, for example, a human cell. So in some sense, we're already creating structures the size of a human cell, which is impressive. However, the caveat is the repeat is only 220 by 200 nanometers. So the goal is, can we build something of this size where it's completely aperiodic? I think that's a holy grail, I would say, for the field of building something of this size, but every single element could be independently addressable. And you could imagine maybe that would allow you to self-assemble memory arrays, or maybe even template nanoelectronics or nanophotonics if you had complete addressable control in 2D or maybe even in 3D over these long length scales. So this is a zoom in 500 nanometer scale bar, and it looks almost like a fabric, like a mesh, like a gauze. So I think it looks kind of neat. One of the things that you might notice is that we have some other structures that are sticking almost randomly on the object. And at this point, we don't know exactly why they're sticking, some kind of non-specific adhesion. We don't know how well we can do in terms of cleaning those off. So that's something that we're trying to deal with right now. But it seems to be an overall kind of random process that we get these extra elements that are stuck like pieces of dirt. So we need to maybe adopt something like the moral equivalent of a clean room strategy or to get rid of those if we so desired. And so this is just to illustrate that, again, these are addressable structures. So what we're doing is we're programming it with specific little handles at particular positions. So this is the 200 by 200 nanometer unit cell. And in this case, we program sticky little single-stranded handles that come out in this pattern. And then we self-assemble something we call a DNA nanocube. It's about 10 nanometers in dimension to hybridize only to the points that we program with the dark dot and not to hybridize anywhere that's faint. So you can see here that indeed we get docking of these nanocubes with the prescribed periodicity of about 220 nanometers. So yes, they're also addressable structures. These are just some different patterns that we created, all with a 220 nanometer periodicity. So here's nice little zigzag lines. You could imagine something like this being used to template nano electronic wires or photonic elements. Shown below here, we're now using super resolution microscopy with DNA paint in order to use light microscopy to resolve below the diffraction limit. And the difference in the intensity of these spots is more has to do with the kind of stochastic nature of imaging. So there's nothing meaningful about this pattern, but we think it looks cool anyways. In this case, these dot positions are programmed with a spacing of about 60 nanometers. So we're using every fourth position here. Okay, so now we come back to this challenge that I gave you in the second slide, which is, can we build finite structures, not just these periodic structures with a 200 by 200 nanometer unit cell? Can we make a unit cell that is the size of the cell? And so I can give you a progress report. So here's the first structure. The red is the seed, gridiron seed below. And we're making a finite structure that's 64 slats, 64 DNA-origamis in size. So this entire structure is programmed from 65 unique DNA-origami. So it's about the same size as the one from Grigory Tikhomorov and Lulu Chen, about 0.5 micron by 0.5 micron in our case. And here's the TEM image. So each one of these top 32 is a different DNA-origami. Each one of the bottom 32 is a different DNA-origami. And therefore, so we fold them in 64 different test tubes or wells, and we could program them with different functionalizations. And then when we mix them together, then each functionalization will know exactly where to go in X, Y, and Z. So it's a fully addressable aperiodic structure. And one of the things that's really impressive about these structures compared to previous ones is the yield at which they're self-assembling. So in this case, we never see anything close to this size or anything that looks like structure unless we add the seed. So it's absolutely seed dependent as far as we could tell. And then the structures pretty much go to completion. It's hard for us to find anything that looks like it's partially formed. It's not perfect. So the individual DNA origami slats, if we calculate the probability that any one of these is there in these structures, we come out to about on the order of 97, 98%. So 2 to 3% of the time, there'll be a slat at a given position that will be missing. So self-assembly is not perfect, but this is pretty good. And we think it's comparable to DNA origami itself, where if you look at the occupancy, so DNA origami is built from a couple hundred staple strands. And when we do tests on DNA origami, we also find that between 2 to 20% of the time, a given staple strand is not there. So I think we can make a strong case that the hierarchical self-assembly of DNA-origami into these structures proceeds with the same kind of robustness and yield as DNA-origami does with individual steeple strand building blocks. So we can get DNA-origami-like yields hierarchically on a much larger scale, and with a seed that is far smaller than a scaffold. Here's a zoom out view with a 6-micron scale bar, and this is just to impress on people that you'll be hard pressed to find too many examples of structures that look like they didn't go to completion, although they'll be missing a strand here or there. And we can compare this with the yield that's from the previous work from Lulu Chen's lab, where they're using a different metric of yield. They're using a more stringent metric of yield, but still, hopefully, you're convinced that our yield is significantly higher. You know, exactly how much higher is not quite clear, but also this is just a progress report. We believe that we can be pushing our yields higher than what we're achieving so far. And so now we're on this trajectory of, can we grow a structure the size and complexity of an E. coli bacterium? Can we grow a structure the size and complexity of a yeast cell? Can we grow a structure size and complexity of a human cell? And then if we ever got there, then we'd of course get greedy, and then we want to build things the size of tissues, et cetera, and then we wouldn't really rest until we had something the size of a human being walking around made out of DNA, and that will take a long time. So we got a lot of work to do still, but so this is back in the spring. We built a structure that is grown from 191 unique slats, DNA-origami. So this was the biggest, by far, hierarchical structure, unique structure that ever been built in the world, as far as we were aware, with dimensions approaching 900 nanometers. This is over 1 gigadalton in mass. So considered very, very large from the point of view of molecular self-assembly. And again, each one of these elements is independently addressable. Each one of these 191 oligomies are first folded in a separate reaction chamber, and then everything is mixed together and they go exactly where they're supposed to go. And since then, we've decided to try to get to 1,000 unique slats, because as organisms with 10 fingers, then 1,000 just sounds like a nice place to stop, at least for the moment. And this is a structure that's only 600 slats. The starting point is here at point 1. Again, it's a bidirectional growth front, and then it comes around and seals. And then this is an individual DNA-origami just for comparison. This is a structure where it's close to 1,000 slats. The dimension per side is almost 2 micrometers in length. So I'm probably going to say this a few times, but an E. coli bacterium is on the order of 0.5 micron by 1 micron. So if we could dock E. coli on the structure, we could imagine docking 8 E. coli like Vienna sausages. So a 2x4 array of Vienna sausages about the size of the E. coli we could actually pack onto this fully addressable structure. This shows the path, the growth path. So we start from the seed, and then it's growing bidirectionally in the directions indicated by this red. This is just a zoom out so we can see. So the yield starts to peter out now that we're making these enormous structures. And currently the yield for these structures that are about 1,000 slats is on the order of 20 to 25%, which is an incredibly good yield considering how absolutely massive these structures are. But again, this is kind of an unoptimized process, and we believe we should be able to push the yield quite a bit higher than this 20% for 1,000 slats. One way to think about this problem is, could we think about the yield per slat? And so you could take 22% to the 0.001th power, and then you come to something like, there's an effective yield per building block of 99.9 something percent. But we think we can continue to do better than that, and we need to do better than that if we wanted to build a structure that was, instead of 1,000 slats, 10,000 slats, or 100,000 slats, or 1,000,000 slats. So this is the biggest structure that we built. It's about 1,022 slats, 2 microns per dimension. You can see that we program it to start its growth in the north pole, and then we get this bidirectional growth front. And we have this nice movie generated by Dio Minev that illustrates the process. So first we have the gridiron seed, and the gridiron seed is programmed to bind 16 unique DNA-origami structures, each one folded in a separate test tube. And then once that forms, then that can recruit the next set of 16 plus 16 slats. So one on the left and one on the right. So we can see what happened here was we had growth from north-south direction, and those now template growth in the east-west direction. And then the same thing on the other side. So it's a bidirectional growth process in this case. One of the things that we're most stricken by is how relatively fast the growth is. So if we measure the second order rate constant for the assembly of these structures, then it's up to 2 times 10 to the 6 per molar per second, which is actually about the same rate that we see individual DNA strands coming together, which might at first sound like, how's that possible that this structure, this lumbering structure that's 300 times the mass of a DNA strand could self-assemble at the same speed? Aren't there diffusional limits that prevent it from associating that quickly? And we're not sure exactly the biophysical basis of this, but I suspect part of it is just that DNA strands themselves don't assemble quite as quickly as they could. So it's not so much that the DNA-origami is doing something mystical. It's just the DNA strands themselves, they have to overcome entropic barriers, and they come together a little bit more slowly than they might otherwise be able to. So we can look forward to, I think, advanced building blocks in the future that maybe assemble in an order of magnitude faster than DNA strands do. We just don't have them yet. Okay, so this is a fully addressable structure. So what we've done here is we've programmed at arbitrary positions these DNA handles to come out so that we can create a pattern. Again, what we're doing is we're docking these DNA nanocubes at those prescribed positions. So the nanocubes only dock at the positions where we programmed complementary handles to be there, and that allows us to get contrast so we can see it using negative stain electron microscopy. So far we've made 4 of these structures that are 2 microns in dimension, and hopefully they're convincing you that yes, they are fully addressable. This is not some random accumulation of junk that just happened to be in the pattern of a smiley face with 2 micron dimensions. Again, E. coli is about the size of a Vienna sausage, half micron by 1 micron. This is supposed to be a puzzle piece. This one is supposed to be a double helix with the Wyss Institute. Maybe you can't quite read that, but you can believe that, again, it's not just some random things that we're interpreting in the wrong way. Oops. Yeah, this is supposed to be the insignia for Harvard University. Yeah, so this is just overview slide from our manuscript draft that we're able to create these structures that are 1,000 DNA-origamis in size. So we can imagine, one way to measure progress is, can we follow a Moore's law type trajectory where we're doubling complexity? In that case, we get double the complexity of integrated circuit every 2 years. So in order to get to something that is 1,000 times the complexity, that should take about 10 doublings. So it should take about 20 years to go from 1 DNA-origami to 1,000 if we just kind of whimsically say we're trying to hit Moore's law pace. So it's been about 15 years since DNA-origami is invented. So you could say we're ahead of pace. We got to 1,000 in only 15 years, so not too shabby. And I just want to say something quickly about the way that we fabricate these structures. It's modular, and this makes it amenable to doing rapid prototyping with relatively small budget. So each one of these 32 handle positions is a different staple strand. And so what we're doing is we're ordering 64 different variants of each of these 32 strands. So that's 2,048 strands total for a budget of, it's a few $1,000s. So you invest a few $1,000s, and then now if you have an acoustic liquid handler, or if you just have pipetters, if you can't afford the acoustic liquid handler, then you could just basically program your pipetting robot to combine these things combinatorially to make any slat you want. So it's like we have a DNA slat synthesizer where at each of these 32 positions, we can put any of 64 handle sequences here at low cost. So the total number of possible slats is 64 to the 32nd power. So that's analogous to a DNA synthesizer where we can put any A, C, G, or T at any position of the DNA strand. Here we can make any slat with any of 64 meta base pairs, if you will, at each of these 32 positions. So it's a quite affordable method once you have the upfront cost of this initial strand library. And so I'd like to argue that what was the secret sauce that allowed us to get this really good yield, even though this is just a progress report, and get to these things that are 1,000 times bigger than a DNA-origami with a 22% yield is still quite impressive for getting to that structure. And I would argue that there's really two ingredients. One is this absolute seed dependence. So you're controlling it. Things aren't just starting willy-nilly in the wrong place. And secondly, this allows you to add a large concentration of the building blocks, and then you can get very robust, rapid, error-free growth. And then just a kind of point of discussion, although I'm going to have to run in about a dozen minutes, is to what extent is what we're doing kind of like a molecular printer? So, and this is another project I don't have time to tell you about. We're trying to literally make self-assembled molecular printers. We have this little print head that we're programming to slide around in two dimensions, and then based on positional control, deposit things onto a canvas. And in this case, the salient feature is that the building blocks that we're trying to position don't themselves have any positional information encoded in them. So it's also kind of analogous to something like translation, where you have amino acids. The amino acid doesn't have any information in it about where it's going in the assembly, but rather it's a template that is encoding the information of where this building block is going. And to contrast that with conventional programmable self-assembly, where we have elements where the information about where this element is going is directly encoded in the building block itself. So you don't need another element to tell it where to go. It knows where to go on its own. And from the point of view of, I just want to manufacture stuff that is of arbitrary shape. One thing I'll just note is that this left-hand path of programmable self-assembly has been undergoing Moore's law type exponential improvements. And this right-hand side of trying to make a print head has been going very, very slowly. So we kind of joke that if things go better than our wildest dreams, then we'll have a printer that can make something the complexity of a DNA-origami maybe 20 years from now. But DNA-origami was invented 15 years ago. And 20 years from now, I will be disappointed if we can't make things 1,000,000 times the size and complexity of a DNA-origami, if not larger than that. And so that raises questions of, well, if the programmable self-assembly is so good, why would you ever why would you want to use this positional approach if it's always lagging behind? And so I can imagine that there could be complementarity between these methods depending on the niche. So the way that I think about it is, one is if you need dynamics, then it could be helpful with an off-the-shelf printer to just kind of move the chess pieces around instead of having to reinvent the machinery to do this dynamics. So that's a case where the printer could still be useful. And secondly, I have this analogy of kind of baking a cake. So if, let's say, the programmable self-assembly is really good at making something the size of a cake, where there's a bunch of interesting features and shapes and nooks and crannies, the size of a cake. But then if you wanted to change some of the surface decoration on that cake, it can be a little bit painful to redesign your process in order to put a new feature in a particular location. So it can turn out to be, you might have to redesign the entire process just to change one little decoration on one part of your cake. So you can imagine a division of labor where the programmable self-assembly makes this sort of generic structure that's more or less what you want. And then you're just using the printer to do some slight modifications here and there. So the printer is like a molecular cake decorator versus, so basically we use this local feature directed growth to grow the cake. And then we just use the printer to decorate the cake. And in that way, both of these modalities could be complementary. Although most of the work you could say is being done by the programmable self-assembly. Okay, so this rounds out my talk, and thanks for sticking with me. So the point is that we have engineered a way to get absolute nucleation control, absolute seed dependence on the nucleation. And we think that allows all or nothing growth, or something approximating all or nothing growth of these microstructures. We think this is a big breakthrough in nanofabrication, and we think it's just the beginning. So we believe that achieving structures that are in an order of magnitude greater in complexity is very near, and maybe something 2-3 orders of magnitude larger, maybe that's not that far off either. So I can stick around for another 8 minutes or so.
Wow.
If anybody wants to ask anything.
This is incredible. Yeah, I think mind blown on every slide. But just to, I guess one question of fun. Do you think that are there any limits to this, like almost like better than Moore's law growth that you foresee? Like, do you foresee it slowing down at one point? Like, are there any barriers in place that you could foresee, or is it just like a free ride, or you just don't know yet?
Yeah, I don't know. I think it'll probably be kind of a bumpy ride. There aren't that many. I think to get a true Moore's law type, very regular trajectory, you kind of need a whole industry. And the industry, basically, Moore's law is not something that just kind of self-assembles on its own. You basically have all these different, as I understand it, you have all these different sub-industries that come up with an agreement. Okay, we're going to do this division of labor, and we're going to split it up in a way everyone agrees we're effectively going to get a doubling of the complexity. And we don't really have that infrastructure, I think, yet with DNA nanoconstruction. Yeah, so one can certainly think about lots of limits of taking the very specific details of the paradigm of we fold each DNA-origami into a different test tube and then we mix them together. There's a lot of physical limitations if you restrict yourself to exactly that paradigm of just how quickly things will diffuse together and come together. But on the other hand, if you say, so a very specific paradigm of growth is going to definitely have limits of how big it will grow. But you can say, on the other hand, if we widen our perspective, we say, okay, we're not going to limit ourselves to exactly this recipe for building these things, because at the end of the day, you and I are built by self-assembly processes. There's no printer that's putting things exactly where they need to be. So living systems are not restricted to just that one paradigm. You have things like molecular motors that can push things around and travel large distances far faster than diffusion can reach. So I don't see why eventually we'll be able to self-assemble. We'll be able to program the self-assembly of things the size of animals, of macroscale animals. Exactly what rate that'll happen, I'm not sure. And but it will require, at each level of complexity, we'll need new paradigms. So I think DNA-origami was a big breakthrough to get to something twice the size of ribosome. I would argue our crisscross architecture is another breakthrough to get us to something that's 3, maybe eventually up to 6 orders of magnitude as complicated. But we're not going to be satisfied with only 1 million DNA-origami complexity. We're going to want 1 billion, we're going to want 1 trillion, and that's going to require more of these technical advances, and hard to predict exactly what those are going to be. But I do have confidence that they will come. People will figure it out. Just a question of when and what will that look like.
Yeah, I mean, sounds like it. I think before we go to participant Q&As, I just want to make sure that we get one question in, which is what we always try to ask. It's like, now extrapolating outward, like what are any potential future applications that you could see from it? And also what can people in this group or people watching this talk on YouTube help to move your work forward? And then we'll go to group Q&A.
Yeah, well, the standard one is resources. So if you know of helpful program managers and things that might want to fund this work, then that definitely will push it along greatly.
And your email is on the website, or people should contact us?
If you do a internet search for me, it won't be hard to find my email address. Yeah, so resources, talented people that might want to work on this. One of the nice things is, maybe, hopefully didn't escape your notice, is that this is quite amenable to automation. So if you have this basically a fixed number of inks, and then you just have a digital control over how those things are rearranged combinatorially, you basically have maybe the building blocks for crowdsourced science. So if we had some centralized facility that just took orders from around the globe, and then we had some more liquid handling robots that basically could self-assemble anybody's design, then that could be something very interesting. If there's some kind of centralized source that could effectively do experiments. And there's some really nice examples of this kind of crowdsourced science in other domains that are quite impressive, in designing RNA structures, designing protein structures, that we could follow that kind of paradigm. That would be interesting. In terms of applications, we're very interested in trying to engineer things like cell interfaces. So we can see examples of cell-cell interfaces that span very large distances that up to this point were not accessible technologically. So if you look at a B cell kissing a T cell to make an immunological synapse, that interface between the cells is something like a couple microns in diameter. And it has a very specific spatial organization where you have this kind of bullseye organization in the middle, where you have this MHC, peptide MHC T cell receptors that are concentrated in the middle. And then you have the adhesion molecules that are peripherally located, and some other things even more peripherally located. And we can start to tease out the language that cells use to communicate over these much larger length scales. As we get to larger lengths, even larger length scales, we can imagine exploiting this for tissue engineering. So of course, there's a lot of interest in being able to regrow organs and tissues outside of living organisms. And so having that spatial control, I think, could be very important for tissue engineering and organ engineering. And then we're also kind of interested in maybe using this for templating nanophotonic structures. So can we position optically active elements to create exotic optical phenomena that require this kind of integration over the micro up to the micron or maybe even larger scale in order to get these very interesting optical behaviors. So those are a couple of areas that we're especially interested in pursuing in the application space.
Oh, that's quite a mouthful already. I will just rush through in the last 2 minutes through a few questions here in the chat. No one has mic, but Stefan Bosley says, really cool structures. Can you start to incorporate functional features too? For example, catalytic units or stimuli responsive elements. Presumably, this could be done with excellent spatial control.
Yeah, absolutely. So anything that anybody's done with the DNA origami, you can do with something that's made of 1,000 origamis.
Okay, great. Then Alex Sturck says, with that degree of control, you could have a block that is functionalized with oriented Feringa motors. Then you could have a raft with an outboard motor, lol.
Yeah, I mean, it's the cells are robots and they're microscale robots. And now we could start to build microscale robots. They could have locomotion, they could have sensors, they could have computation. Yes, it's to a large extent, it's a question of complexity. And we haven't had access before because you need these breakthroughs to get to greater complexity to avoid those errors taking over. So self-assembly is always error prone. And the key is at each level of new complexity we're trying to achieve, can we reduce the error rate by another couple of orders of magnitude and get to that next level complexity? But then we're stuck again. And we need some more breakthroughs to get through those errors. And that's basically how this is going. It's a series of steps, tricks to lower the errors, not to zero, but just lower the error rate tenfold at a time.
Okay, I'm asking the other one without Mike. How strong are the final structures? Do they fall apart at the slightest touch, or do they seem to hold together pretty well relative to, say, a cell?
Well, we haven't tested that extensively, but they can survive centrifugation. So if we spin them down, we can get them to sediment and we can resuspend them. Yeah, so that's an issue as you grow these large structures. Two things. One is their mechanical stability, and second is just how sticky they become. You have this large surface area. And so there's some common sense things that one can approach to try to address the strength. So one can do cross-linking, one can try to coat it with other materials, one can try to build with the geometry that is more resistant to shear. You can coat, you could imagine microparticles, you could coat one of these things, you can drape it over a microsphere, so you can impart the strength of the microsphere and make it an addressable microsphere. So there's a bunch of these kind of common sense kind of things that you can start to do to try to deal with that.
I like how you say common sense there. Okay, do you have to hop off like right at the hour, or?
Yeah, I should probably sign off. But it's been a lot of fun to present to you guys. We hope to submit to a bioarchive in the next few weeks, and then maybe at that point it would be okay to post this online. And if you have anybody wants to engage further, I'm happy to talk more. Okay, especially if you guys have ideas of resources that we could add to this exciting direction.
And I know your time is like, you're super busy, and Dionys is also, but like we have now one of your researchers who you mentioned earlier in the presentation as a Foresight fellow. So maybe we can also direct some of the questions to him if people have questions.
Yeah, he's really awesome young scientist. Couldn't have done any of this without him.
He looks, his work looks fantastic. Thank you so much for taking the time.
Okay.
I will coordinate with you when we can post the talk. Thanks a lot. This was fantastic.
Thanks, everyone. Bye-bye.
Thank you, Will. Bye.